Hello everyone, my name is Tayfun Tatar. I'm an application scientist at HansaBioMed Life Sciences, and today I would like to walk you through the frontiers of extracellular vesicles field pioneered by HansaBioMed Life Sciences. Before I start my presentation, let's have a look at what we will cover during this presentation. So I will first start with an introduction to HansaBioMed Life Sciences, which covers what EVs are as well as what we do as HansaBioMed Life Sciences in the field of EVs. But then I would like to dive deeper into bestsellers of our product portfolio, namely tangential flow filters and EV standards. So now let's start with an introduction into EVs. EVs are the cells' way of communicating with each other, and they regulate intercellular communication by the help of the functional molecules like proteins or mRNAs that they carry within their cargo. They can be classified based on several different criteria, and these include their sizes, cellular origins, cargo, or even cellular function, and they are available from many different sources. As HansaBioMed Life Sciences, we are active in the field of EVs both commercially and in the research settings. Our commercial involvement includes, first of all, product development in the EV field that covers purified EVs, EV isolation tools, EV characterization kits, RNA isolation kits, antibodies that are used in EV research, and liquid biopsy tools. But we also offer custom services to our customers to ensure end-to-end EV workflows in a successful way. In the meantime, we are also actively involved in several research projects to pursue innovation in the field, and here you can see only a short list of our past and present research projects. In order to give some better sense of our commercial activities, as for the product lines, you can see that we are mostly selling our purified EVs and isolation tools, which I will cover a bit in more depth shortly. And as for sales regions, you can see that even though we are much more active in North America and Europe, we are actually serving EV researchers in a global manner. And in order to give some more details about our bestseller products, on one hand we have EV isolation tools, and we have a variety of EV isolation tools because EVs can be isolated by targeting different features, and we would like to offer the researchers the ideal method depending on their use case. So for this reason, we have TFFs for EV concentration, purification, or size-based separation. We have size exclusion chromatography columns that can be used for EV purification, or we have centrifugal size exclusion chromatography columns that are ideal not only for EV purification from minimal sample volumes, but also for unbound dye and antibody removal. On the other side, we have purified EVs that are so-called EV standards or EV reference material, depending on the context. These are basically EVs purified from cells, human biofluids, plants, bacteria, fungi, or any other sources, depending on the request. And they can be used for assay calibration as positive control, for instrument calibration, namely as standards in short. We also have a first of its kind product that is called Fluo EVs. Fluo EVs are the product of genetic modification done to their parent cells, namely human embryonic kidney cells. As a result of this genetic modification, these EVs carry EGFP, PFP, or M-cherry fluorescent proteins in fusion with the well-known EV surface markers CD9, CD63, or CD81. This way, without any external labeling step, these EVs are stable fluorescent, they don't bleach, or they don't cause non-specific labeling caused background signal. Hence, they are ideal for fluorescent mode of measurements for assay calibration or instrument calibration especially. As for our services, as I mentioned earlier in my presentation, we would like to offer our customers an end-to-end workflow as a solution for their EV-based needs. This starts with purification of extracellular vesicles in a scalable way that can be applied to any EV source, namely biofluids, conditioned media, and plants. And of course, this purification comes together with validation that follows MySav standards. Then we can also perform phenotyping and imaging of EVs with various technologies, including NTA, TEM, ELISA, or flow cytometry. For purposes requiring enrichment of specific EV subpopulations, we can also perform such tasks, especially if it is required in diagnostic or therapeutic settings for biomarker discovery. And we can also couple all these with EV omics services that include proteomics, transcriptomics, genomics, lipidomics, and metabolomic analysis of EV markers, coupled with high-level bioinformatics analysis. While we are performing all these activities both in commercial settings and in research settings, we also pursue several partnerships and collaborations both in academia and in industry. Here you can see only a short list of our existing or past collaborations, and we are also open always for further collaborations. After this summary, let's have a closer look into our tangential flow filter cartridges. But before that, I would like to start with an introduction into what tangential flow filtration is. Tangential flow filtration is also known as cross-flow filtration, and it is a method based on filtrating the sample in a perpendicular direction to a laminar flow. It is a widely used technique in biopharmaceutical or food industries, and its main uses cover purification, concentration, or defiltration of biosamples. In more technical terms, tangential flow filter cartridges or hollow fiber filters include multiple submillimeter diameter fibers made of non-binding materials. This can be polyether sulfone or polysulfone, and all of these fibers have a set pore size. When the sample is subjected to a laminar flow within these fibers, the particles that are inside the sample, which are smaller than these pore sizes, are filtrated out, whereas the particles larger than this pore size remain inside. So in its essence, tangential flow filtration is a size-based separation method. In practice, you simply use our TFF cartridges by injecting your sample within syringes that come with luer lock ending. Once you filtrate these syringes back and forth until there is no filtrate coming out, you basically separate your sample based on the pore size, and depending on the application, your sample is either what retains inside the filter or what is filtrated out. So in its essence, as you can see, tangential flow filtration is an ultrafiltration method that separates particles based on size. But what makes it different or more advantageous than dead-end filtration is that one of the critical challenges in dead-end filtration is the accumulation of particles larger than the pore size over the filter in the case of dead-end filtration. Because such particles that are larger than pore size accumulate over the filter, growing a cake over time, and this decreases the flow rate. In tangential flow filtration, on the opposite, the laminar flow to the perpendicular direction of filtration ensures cleaning effect on top of pores, and this prevents cake formation or clogging of the filter, ensuring a steady filtration. Of course, it is safe to say that TFF is not a technique that is invented for the field of EVs. In the opposite, TFF is actually invented during 70s or 80s as a method for protein purification and cell separation used in research settings. But as it is matured more and more, it is introduced also in other fields, and it was only in late around 2010s that it is started to be embraced by the EV field as an ideal EV isolation method. And ever since its recognition by the EV field, you can see that the number of publications done annually by tangential flow filtration as an isolation method grows almost following an exponential trend, and we can expect an even sharper increase in the number of TFF based EV isolation after this point, because in the beginning of 2024 TFF is recognized as an ideal method for EV concentration in the MySav guidelines. But what are the advantages of TFF that make it so useful for the field of EVs so that it can be embraced so quickly? So, as in any other settings, the best thing to do when you are introducing a new method into the field is to compare it with the gold standard. And for EV isolation, the gold standard is ultracentrifugation. So if we compare tangential flow filtration with ultracentrifugation, first of all, ultracentrifugation exerts high centrifugal force on top of EVs, and this causes the particles to become sticky or even result in a fractioned membrane, whereas in tangential flow filtration, the smooth flow of particles ensure that there is no aggregation. Ultracentrifugation can provide only a poor recovery of EVs that is limited to around 30, 35, maybe 40%, whereas tangential flow filtration can exceed even 80% of recovery. Ultracentrifugation is a time-consuming method that requires hours to provide you with your EV samples, whereas in tangential flow filtration you can process hundreds of milliliters within only minutes. Ultracentrifugation is not a suitable method for scale-up, which is a limiting factor especially for industrial settings, whereas tangential flow filtration ensures easy scale-up from 5 milliliters up to tens of liters. Ultracentrifugation, since it doesn't have a clear threshold for isolating or purifying EVs, it contaminates EV samples with other particles, whereas in tangential flow filtration, the sharp cutoff inserted by the pore size of the fibers minimizes the contamination. In ultracentrifugation, there is a requirement for expensive lab equipment, whereas tangential flow filters can even be used manually, or if you want to automate it, it can be done so only with a peristaltic pump. But of course, it wouldn't be fair to claim that TFF is advantageous only when compared with ultracentrifugation, because actually it possesses serious advantages over many isolation techniques used in the field of extracellular vesicles. And these advantages mainly come with the versatility of TFF, which allows it to be utilized in various different scenarios, such as separation and isolation of small or large EVs, concentration of pre-isolated EVs, buffer exchange or dialysis, removal of contaminants such as unbound dyes or antibodies, EV depletion from FPS, or even elimination of unwanted proteins such as THP in the case of urine. But the possibilities that you can explore with TFF is not even limited to the advantages that are brought by its versatility, because you can even increase the degree of exploration by combining multiple tangential flow filters in series, as you can see on the schematic view on the right. So this is a small study that we have done in-house by combining tangential flow filters with 200 nanometer pore size, namely TFF MVs, and then we used its filtrate as an input to TFF EVs with 50 nanometer pore size, and this way we were able to collect EVs sized between 50 to 200 nanometer size from various plants. But then the degree of exploration is not even limited by combining multiple tangential flow filtration cartridges in series, because you can even combine multiple methods with them, especially orthogonal methods such as size exclusion chromatography or anion exchange chromatography. And in such combinations where you need higher purity for the cases like plasma or serum, the advantages TFF brings into play are mainly concentration of EVs, because TFF can start with hundreds of milliliters or even liters, tens of liters of volume, and concentrate the sample down to very small volumes that these chromatography columns can work with, and it can also remove small solutes as well as large debris simultaneously in a scalable and efficient way. But even more possibilities can be explored now with our new automation system that you can see on this image, because by the help of this system now you can do all of these versatile approach, different scenarios in an automated and controlled way by the help of its integrated software, and by minimizing human intervention. This automation also increases the reproducibility of your results, which is ideal case scenario, especially for industrial settings. After revising the technology behind it, as well as the advantages of tangential flow filtration for the field of extracellular vesicles, now let's consider some real case scenarios where our tangential flow filtration cartridges have been useful for EV researchers. In the first example, the idea of the researchers is to compare the immunomodulatory and regenerative potential of EVs from induced pluripotent stem cells versus human umbilical cord derived mesenchymal stem cells. And in this process, after isolating EVs, they use our TFF Easy with 5 nanometer pore size in order to concentrate the pre-isolated EVs down to the required volume, where the dosage of the injected EVs are a matter of concern. And by the help of such studies, induced pluripotent stem cell derived EVs were indeed shown to possess higher pro-regenerative potential through in vitro scratch assays. In the second example, now the starting biosource is bacterial outer membrane vesicles, which promise a platform for vaccine development as well as drug delivery. However, the challenge included in this is that measuring their biodistribution is very cumbersome, and the aim in this study is to come up with a high efficiency radio labeling method for in vivo imaging studies. For this process, after isolating outer membrane vesicles from bacteria cultures, they again used TFF Easy with 5 nanometer pore size to concentrate the isolated EVs before injection into mice. Again, a case where dosage is quite important, and by the help of these studies they were able to come up with an efficient method for radio labeling in vivo. In the third case, now the starting material is another biosource of EVs, which are plant EVs or citrus EVs to be more precise, which present an excellent platform for nutraceutical delivery by the help of their endemic cargo, which includes multiple micronutrients. However, the problem is that their cargo is inherently unstable and it is affected by different storage and preservation conditions. And here the idea of these researchers in this study is to come up with an optimized protocol for storage and preservation. For this purpose, they now use two of our tangential flow filtration cartridges, namely TFF MV with 200 nanometer pore size for isolating EVs from plant extracts, and then TFF Easy with 5 nanometer pore size for concentrating the EVs after isolation. And through these studies, they evaluate physicochemical characteristics and antioxidant activity of EVs isolated with this way after storing and preserving in different conditions. And in the end, they were able to come up with an optimized protocol for preserving and storing EVs. Now in the fourth example, the starting material is conditioned media, where the hypothesis or the question of research is the role of EVs in human limbal niche homeostasis, which is still poorly understood. And here we see a combinatory use of tangential flow filtration in combination with size exclusion chromatography columns for further purity. So this is also showing that combination brings further advantages to the researchers. So here, after collecting the EVs through TFF easy with 5 nanometer pore size from conditioned media, the researchers performed further proteomics analysis on top of these isolated EVs, which resulted in the conclusion that EVs from human limbal niche may be actually regulating keratinocyte development, extracellular matrix organization, as well as niche regulation, which might be their role in the human limbal niche homeostasis. And then in the last example, now our starting material is bovine milk, which represents an abundant, cost-effective, as well as eco-friendly source of EVs. And it also demonstrated a promising potential for delivering chemotherapeutics as well as bioactive natural compounds. However, the challenge that yet remains is the scalable production of bovine milk EVs, which requires an optimized protocol. And here the idea is to come up with an optimized protocol. And the optimized so-called protocol in this study by these researchers, especially for the industrial settings concerning the scalability, includes enzymatic casein removal via rennet, which is an enzyme that can capture casein, followed by tangential flow filtration processing. And now, after completing the part related to our tangential flow filter cartridges, now let's move on to another bestseller of our product portfolio, which is our EV standards. So for this purpose, I would like to start with giving an understanding of who would need an EV standard. So as a dynamic field, the field of extracellular vesicles stages continuous improvement or research for improvement on EV-based therapeutics where extracellular vesicles are to be used as a drug loading agent, on EV-based diagnostics where EVs are preferred as a source of biomarker, or even development of new methodologies, new approaches for detecting, characterizing, or analyzing EVs. And in order to integrate new assays, new methods, or new instruments regarding all of these approaches, you would need a reference material, a reference EVs for validation of your approach. And here in this video, now you can see the growth in the number of different characterization techniques that is used in the field of EVs, starting from 2000. As you can see, over the years, it is not only an increase in the number of papers using each different method, but also new and new methods year by year come into the field of EVs. Of course, some come in a much more dominant way, some are used much less frequent, but this doesn't still prevent us from seeing that there are more and more techniques that are used over the years. And in order to validate each of these methods, you would need an EV reference material, as I mentioned earlier. In order to respond to this need, at HansaBioMed Life Sciences, we are providing EVs isolated from various different sources, namely biofluids, conditioned media, or plant extracts. These are isolated with tangential flow filters and further purified with size exclusion chromatography columns or anion exchange chromatography in the cases where it is necessary. And we further lyophilize these, which enables them to be stable for long times and to be shipped without dry ice conditions. But of course, once you claim that you are commercializing EV standards, the first question that comes is, are they really EVs? So in order to convince or in order to demonstrate that these are really EVs, or in order to validate that these are really EVs, we are following MySav guidelines, which are widely accepted in the field of EVs. And MySav guideline basically states that EV characterization should include size and concentration analysis, which we perform by NTA measurements, morphology analysis, which we satisfy by TEM analysis, and then marker analysis, which we perform through ELISA analysis through 3 membrane markers, 1 internal marker, and then 1 negative marker. And also we confirm the purity of our samples, because it is not only enough that there are EVs in your solution, but these EVs should be as pure as possible. And for this purpose, we basically take the ratio between the number of particles as measured by NTA and the total protein content as measured by BCA. And this is basically a ratio suggested by a paper that is widely accepted in the field, and the publication date was years ago, so it is now widely used. And this way we can ensure our audience that we have instant EVs per se that they can use in countless use case scenarios. And here I would like to introduce you to some examples. So in this first example, the idea is to come up with an EV functionalization method, because the current methods available for EV functionalization suffer from efficiency and standardization. And here the approach that is used is an enzymatic engineering way to glycan regions, which enable surface functionalization. And for this purpose, in order to show that their method for EV engineering, EV functionalization is versatile in the sense that it can be applied to multiple biosource of EVs, they utilized EVs from serum and multiple cell conditioned media, and they demonstrated the applicability of their approach in various EV types. In this second example, now the focus is on cancer therapeutics, with the idea that despite there is a growing interest in novel cancer therapeutics approaches, therapeutic monitoring is still challenging, and the idea is to use a quantitative approach of EGFR, epidermal growth factor receptor, that is on EV membranes to assist in monitoring. And to show the feasibility of this approach, the researchers used extracellular vesicles isolated from HCT116 cell line, which is a colorectal carcinoma cell line, in their assay development. And during their studies, they were able to demonstrate that this approach on quantifying EGFR holds promise not only for diagnostics but also for therapeutic monitoring in EGFR-driven malignancies. Now in this third example, the focus area is now measuring EV distribution, EV biodistribution, because EV biodistribution is a critical aspect of EV therapeutics. You need to be able to measure or ensure that the drugs reach the target area after injection, and biodistribution is a critical aspect for this purpose. And this study focuses on measuring this for critical therapeutic applications by using EVs derived from HEK293 cell lines, which is human embryonic kidney cell lines. And this way they were able to develop a qPCR-based assay targeting mitochondrial DNA to measure biodistribution. Now in this example, the focus area is a bit different than the ones before, because this is now not an assay development or not focusing on therapeutics, but this is now a method development, or that can be considered as an instrument development, with the idea that for EV-based diagnostics, much more rapid and sensitive methods are required than the ones currently available in the market. And this method, aiming on this purpose, is developing on light-induced acceleration of specific binding between the EV surface and the antibody-modified microparticles through a controlled microflow with 3D analysis that is performed by confocal microscopy. And in order to demonstrate that this method can indeed detect or characterize EVs, they use different EVs from two different cell lines, namely HCT and A549 cell lines, and they demonstrate by the help of our instant EVs, our lyophilized EVs, that their system can actually detect 10 to power 3 EVs in 500 nanoliter samples within only 5 minutes. Now that we are considering the characterization aspect of the extracellular vesicle research in general, it is also a good point to consider that due to the nanoscale of EVs, most analytical methods that you can use for EV research are based on fluorescent approaches. But fluorescent labeling comes with many different issues, like stability of the fluorophore, bleaching, or even non-specific background signal. That is the case especially with rich samples. And we aim to solve this problem by our fluorescent, stable fluorescent EVs released from genetically modified HEK cells. By the help of this genetic modification, our fluo EVs, as you can see in the representative image on the right, carry EGFP, BFP, or M-cherry fluorescent proteins in fusion with the common markers of EVs, CD9, CD63, or CD81. By the help of this genetic modification, these EVs are stable fluorescent, they don't bleach, or they don't have non-specific background signal because you don't need external labeling. And this makes them an ideal product for assay validation or instrument validation that require fluorescent mode of operation. And here you can see some examples where it can be used in a versatile way in multiple scenarios covering multiple technologies. On the left you can see that it can be used as a reference material in super resolution microscopy measurements. On the right you can see that they can be used as a positive reference material also for nanosorting. And here on top left you can see that they are also employed as a proof of concept generation for Taylor dispersion analysis based size and concentration analysis. On bottom left you see that they can be also employed for in situ labeling and quantification in complex media by microfluidic resistive pulse sensing technology. And finally, on the right, you can see they are also an ideal reference material for nanoflow cytometry measurements. And finally, as part of our EV standards product line, I would like to mention plated EVs, which is one of the newest addition to our product portfolio. So here our idea is that plasma EVs are considered one of the high interest areas of therapeutic research and applications when it comes to EV research, but their challenge is that more than 99% of the nanoparticles present within plasma are lipoproteins, which have more or less the same density and size range as extracellular vesicles. So it is quite cumbersome to get rid of as many lipoproteins as possible within EV present solutions. And even though you apply isolation and purification, you can only enrich EVs. However, when you consider platelet extracellular vesicles possess very similar therapeutic capabilities to their plasma counterparts, and they are much less contaminated with lipoproteins, and this sets them forward to their plasma counterparts for therapeutic applications based on extracellular vesicles. So considering these advantages, now we also have platelet EVs available in our product portfolio, both in lyophilized and frozen form upon request. And finally, before closing my presentation, I would like to mention that even though I focused mostly on our tangential flow filtrations and EV standards as two of our best sellers, I would like to mention that we have much more options, much wider product portfolio for EV researchers, including size exclusion chromatography columns, centrifugation ultrafilters, spin columns, precipitation reagents, immunoaffinity isolation tools, as well as characterization kits. And we even offer EV research services to our audience. If you are interested in further reading for any of the papers I mentioned during this presentation, here are my references. And if you have any questions, please feel free to reach out to me whenever. And otherwise, thank you for your attention.